Good evening to all the participants. It is my privilege to introduce our esteemed speaker for this session of Faculty Development Program. We have with us Dr. Rajitha B, an accomplished academician and researcher currently serving at the Motilal Nehru National Institute of Technology, Prayagraj. Dr. Rajitha has an illustrious academic journey. She completed her B.Tech in Computer Science and Information Technology from Kamala Institute of Technology and Science, Telangana. Pursued her Master of Engineering in Computer Science and Engineering from Karunaya University, Tamil Nadu, and earned her PhD from MNNIT, Allahabad. With over 14 years of teaching and research experience, Dr. Rajitha has made notable contributions in diverse areas including image processing, medical image processing, multimedia processing, machine learning in computer vision, deep learning, and quantum machine learning. She has taught a wide range of courses such as artificial intelligence, data science concepts and methodologies, software engineering, image processing, and introduction to AI/ML. Her expertise and passion for teaching and matched by her strong research credentials. Dr. Rajitha is also a senior member of IEEE and a member of ACM, reflecting her active engagement with the global research community. We are truly honored to have her with us, and we look forward for gaining valuable knowledge and perspective to her session. The session is your ma'am.
Thank you, Dr. Abhishek Mishra, for your kind welcome. Sir, should I start my presentation then, the talk?
Yes, ma'am.
Okay, so I'm actually sharing my screen. Let me know if it is visible or not. Is my screen visible now?
Yes, ma'am.
Okay. Okay. Thank you so much for your kind invitation in your university for this online workshop. And myself, Dr. Rajitha, as Abhishek sir has introduced me, I'm working as an associate professor at MNNIT Allahabad since 2012. And my main research interests include image classification, computer vision, medical imaging, and right now I've just started my research in quantum processing as well. So today's my topic would be in the future of medical image classification using the deep learning models and the how we can deploy or how we can have the quantums as well for deploying the classification models in real time. So let us focus on our topic. And in today's topic, today's talk, I would be talking about the few basic things here, like what is medical imaging and what is an image, types of an image, types and images. What type of images do we have in real time applications? Then we will be looking at importance of medical image classification, which I'm actually taking into a short notation as MIC. I'll be calling as MIC now my slides, medical image classification. And then we'll be looking also at what are the current challenges. And then we'll be looking at motivation of using deep learning, followed by what all methods we have adapted in deep learning in medical image classification using the deep learning models. Then followed by I'll also be talking about few challenges what are left out even with deep learning models, wherein the rise of quantum might help in doing the better classification of medical images for real time adaptability. Okay, now let's look at the basic. What is an image? Now, as we all know, an image is like a picture or pictorial representation where human can look at any description or any natural scene. Now, if I talk about a digitized image, the natural scene or the captured image should be stored in a machine in a form of digitally. So, a digital image can be defined in terms of mathematically as a two-dimensional function noted as f of x comma y, where x and y where x and y are the spatial coordinates, and we will have an amplitude f at a pair of coordinates xy, which is called in image is to be as an intensity of that level. And these intensities are having the property of discrete quantities. They are not continuous because you are having an image which is having a pixel, and pixel is in the form of an numerical value, so it is always a discrete entity. So now that pixel we are calling as an pixel. Now pixel is uh, image. We didn't represent the image in the form of an like digitized image which we are storing in an particular device. So how do we capture that image? As we know, we will be having a camera which will be projected on some object through the light illumination whatever we have the energy source. That a real-time object is captured through this device into a digitized photograph. Now, this digitized photograph is stored in your machine in this form, like it is shown in the last one, which looks like a 2D matrix. A certain rows and columns followed with an every pixel cell. Now, this every cell is represented as an pixel, right? Now this pixel can be of holding different values, due to which your color gets generated in your image. Now basically the basic type of image is an 2D matrix which we are storing to capture the original object representation. Now, this particular pixel or the image is taken in from the device, a camera. Now, that camera will be taking a spectrum range of visible light spectrum from 400 nanometers to 8750 nanometers. So, the image will be generated within this particular range. Now every device should be capable, every device what we are using for digital image generation, it uses this particular range itself. Now based on, if you can see this the screen which I'm showing right now, if you look at the different spectrum ranges, now depending on that frequency level, the type of image what we what gets generated is different. Like some gamma rays can produce different type of imaging, x-rays will produce different type of imaging, ultraviolet rays develop different type of imaging, radio waves and infrared rays will produce different type of imaging. That means we will be having different types of devices to capture different forms of images in real time. Now every device is not the same. Now categorization is done according to the what wavelength spectrum it can hold and what type of images it can create or capture. Now depending on which we have wide varieties of images, which of them are certain examples are given here. Few of them is the gamma rays can produce these type of images as I'm showing on the screen. Similarly, the X-ray spectrum can produce the X-ray of an human, of an organ, and or like an digital scan of an X-ray, like we do at the airports, right? So we have a different spectrum for different type of application. Now, X-rays are used for different form of applications. Gamma rays are used for different form of applications, and ultraviolet are used for different sorts of applications, and visible light spectrum is used for different type of application. So like this we have n number of images which are generated in real time. So the radio waves can be used to generate your MRI images, infrared rays can be used to generate the thermal images, and similarly synthetical images can be generated using the microwaves, right? So like this we have wide sets of imaging, and which can be captured or which are captured for different sets of analysis in real time, or the observations which we can be doing in real time. So, similarly, ultrasonic imaging is also one such category, wherein we try to look at the internal organic function of an body, of a human body. the as you can see from all these slides, like around 3 to 4 slides, you can see that all the data that we want to capture, which is not visible by a normal human eye, can be captured with different sets of rays. Now, you by using different sets of devices which can generate those waves, you can generate different sorts of images. Now, our topic is more specific towards the medical imaging. Now, I'll be focusing only on medical domain, not the other real-time computer vision and other types of applications. So, your others today's topic is specific to imaging, so we will be looking only at medical domain applications. Now, if I try to store an image in a machine, as we have already seen that the image is stored in the form of f of x, x comma y in a 2D matrix. Now, I told you that it will be stored in the form of a matrix. Now, how does it store? How does it really impact in the memory? If you can see the particular slide which I'm presently presenting, here you can see that the image what I'm going to hold here is of size 2724 with 2 through 2336. Right now, these many is total at is the total number of pixels. Now, under this, if I look at a certain block, a certain smaller block that is arranged in this form again. Right. So you have the number of intensities here 82, 83, 81, 70, 77. There are wide number of values. Now, what are these? These are called the intensities. Now, these intensities represent the color in your image. Now, that color would be a darker or blackish, whiter. So it depends on the type of image. Now type of in the image in the sense that either it is a black and white, is it a grayscale, or it is a color spectrum image. Now, for black and white, the intensities will be generally between the number of bits what is used for each pixel storage. So, if it is generally an 8-bit image, then we will be having the pixels in between 2 power 8, that is 0 to 255. So, 0 indicates a black pixel and 255 indicates an black white pixel. So, like that, the pixel organizations are being framed for generating an visualization of a real time object. Now, this image is generally a 2D discrete values. Now, this image, how it get generates, how it is stored, depends on the device what we are using for capturing it. So, like if I talk about X-ray images in general, most of we generally we take the X-ray imaging when we have a bone, when we have to analyze the bone structure. Is there any crack, or is there any swelling, or is there any bone tumor or something, then we have to look out for X-ray imaging. Then we can see that the X-ray images are generally like black and white, mostly like black and white shades. Now, does it mean that it is a black and white? No, it's not a pure black and white image. It's actually a form of color only. It's not even a grayscale image. It's a color image only, represented in the form of RGB. Right. So now that actually generates only in the spectrum. There is no color spectrum in gamma rays. So we have the color shades in the form of a blacks and grays in a combination. Right. Now, but it is having a three different channels, RGB. Right. So the one which I'm presenting on the screen, it is simply a 2D image, which is we call in practical as an grayscale image. But when I talk about the color image, generalized images of medical domain, like we can see in the current slide, the medical domain will have lots of data in different formats. Not only imaging, there are different data formats like genomic data, ECG data, radiological data, cardiac catheterization, echocardiogram, blood pressure data, CT scan report, and many. Right. So, like this, we have different forms of medical data. Right. Now, under this medical data, what we have, if we have to do some classification, if we have to do some prediction, and if you have to do any clustering or something, then you need to know the respective properties of that data category, whatever we are using in. So suppose if I try to work on medical imaging domain like radiographical data or CT scan data, then we need to look out or understand how the image is stored in your medical domain, and what are the different forms of data storage or data creation in real time. Similarly, if I talk about genomic data, it is like a character set data set, right? There is no nothing about pixel or something. So the entirely data set is different. And similarly, ECG looks like a signal data. Blood pressure is actually again some numerical values which we are trying to predict or which we are going to have as an outputs of blood pressure data analysis, right? So now if I look at this different types of applications in medical domain, every device produces different forms of information, and in a different format actually. So now you have different models to train them, and different types of models you can use for respective classification. Like ECG classification, you need to use a different model structure. Blood pressure model data classification, you can use a different data models. Genomic data classification, you can use different set of classification models. Similarly, for images we can use different sets of classification models. Now our title is comprising two medical image classification, so we will be talking about what type of medical images we can have, how many ways of formats or you can store, and what are the different types of medical imaging we have in general in today's scenario, and what type of models we can adapt there, right? So now if I look further, this slide presents different types of medical domain itself. I'm not talking about generalized images. Under medical domain itself, we have wide sets of collections of images like X-rays, wherein we get the X-rays of radiography, mammography, fluorescency, and so on. Like this, we have different sets of applications where we can use X-rays. Similarly, ultrasound imaging, which is used for understanding 2D ultrasound, 3D ultrasound, and Doppler ultrasound. So the images are different from X ray to ultrasound. Similarly, we have CT imaging, which is for angiography, and so on. Similarly, the widely adapted or widely research used application is the MRI data and X-ray data, popularly used. These two data sets are widely available over web, wherein many researchers have done lots of work on that depending on the type of disease they are focusing on. So MRI is magnetic resonance imaging, which has MRA, MRP, and FMRP. Like this, under MRI also you have wide sets of collections of imaging. Then we have PET, which is like positron emission tomography. Now, similarly, we have SPECT imaging, which is like single photon emission CT, DSA, OCT, endoscopy, thermography, and digital pathology. Like this, we have different sets of imaging modalities in medical domain itself. In medical domain, right? Now, all these imaging will produce different sorts of information about your same body organ which we are targeting on to identify some disease or to predict some disease or a daily regular analysis whatever we want to perform. We can use the respective type of modality to do the medical diagnosis on that particular patient. So now, since our topic is more specific to only deep learning models, so I'm not focusing much on the other models. But if I said in one sentence, if I say what all the data we have, either X-ray images or MRI data or PET data or whatever medical imaging we have for every disease, whatever we are talking of, if any disease is there, disease D. Now this disease D data set first we need to collect or we have to find out or we need to get from the respective hospital. Now this data will can be used for analysis in three analysis for various applications. One of the application is classification, and another application is prediction, right? Like in classification you have already a pre-trained model or pre-trained labeled data where you are taking the existing diseases, already you have a knowledge of with you, and then your now new disease or new patient comes into the hospital, you can classify depending on the symptoms, depending on the feature set, depending on the body organ data, x-ray image or ct image or mri image, we can classify whether the patient belongs to which type of disease. Then accordingly the diagnosis can be done. Similarly, for predictive analysis also, we can use the same data set and do a predictive analysis. And the similar type of data can be used for clustering as well. Matlab, for identifying the groups, similarities of different diseases. Like different diseases might have 80% similar symptoms, but 20% similarities differ between them, right? And there are many diseases wherein you have different symptoms entirely. Like that we can get the data. If we don't know anything about the disease, then we have to look at the internal relationships among the given samples. That means the given data set, whatever is given to us, looking at the images or looking at the pathology report or looking at the clinical report analysis, we can classify them, or you can tag them into the respective disease. So this comes under the unsupervised approach, where we don't have any real information, and then we are trying to identify the among the relationships within the given data itself. Like that, we can use this medical data for clustering, prediction, and classification. Now, when I talk about these data sets in real time for classification, now classification, if I talk about specifically about classification, now classification can be using basically two types of approaches. One is supervised approaches and reinforcement learning approaches, right? Supervised, unsupervised, and reinforcement. Unsupervised completely comes under clustering, so that is why I haven't labeled here. So in classification you can use supervised approaches where you have already the data, and we can also use certain in advanced sets of applications where you don't know anything about the disease, because you can see that now these days the diseases are growing widely. There are new diseases getting generated where we don't have any labeled data, labeled sample with us, right? So we need to predict based on the new symptoms, new data set what we are getting, new relationships what the patient is going through, what are the new symptoms the patient is going through, looking at those relationships and the behavior of the patient, how he is behaving, what is the effect due to that new symptom in that human body. By looking at all these real time replications on a human body due to some new disease which is not detected already in earlier, there was no history of such disease, then the learning what we can adapt is the through reinforcement learning models. So where we don't have any prior knowledge about the model, right? So in classification, if I talk about, it is either the full-fledged the labeled data is given to us, then go for supervised. Like if we don't have any labeled information about the new disease, then we have to adapt reinforcement based models for detecting or for classification of new disease. Now, if I talk about this, say either the supervised models or reinforcement models, first let us look at supervised models only. In that, we can use either machine learning models. These supervised models can, the data can be supervised, it can be used a machine learning model. Here we can also use a deep learning model, or else we can latest we can say transformer based models, learning based models, right? So under labeled data, we have a different sets of collections of diseases already labeled. We know the symptoms, we have a clinical data, we have a medical data, x-ray image data, let's say ct or whatever the imaging data we have clinical report, we have pathology report, we have a dna sequence report, everything we have with respect to one disease of a patient. Now this whole multimodality data we have, in this we can work either on single modality or we can work on multimodality. So now if I'm in both of this, whether if I'm just targeting on medical images based classification model, then it is called a single modality model. Now, under this, if I'm considering a patient's medical image plus the same patient's clinical report, the same patient's pathology report, the same patient DNA sequence report. Suppose like this, if I'm for the same patient X, I'm taking 4 modalities of data like X-ray and CT scan or something, X-ray, clinical, and pathology report, and followed by the DNA sequence report. Now, if I consider all of them and generating a machine learning model to train it, or I'm generating a deep learning model to train it, or else I can go for transformer based model to train it. Right. So, like this, I can use 3 different models on same modality of data for detecting a particular disease Y, or I can consider different modalities of data of same patient and use machine learning, deep learning, or transformer learning model to decide whether the disease is Y or not. Right. So, like this, I can adapt similar modality or different modality on 3 different ways: machine learning, deep learning, and transformer based models. So now if I talk about machine learning models, now these machine learning models already you are already under looking at for from 4 days, you are already listening to most of the lectures. By then I think you already knew that what is machine learning and deep learning. Now, machine learning is something like the models, if I name, like SVM, random forest, naive Bayes, neural network also you can apply, like artificial neural network is also can be adapted in machine learning. Like this, we have a different ADA boost, XG boost, and so on. Like this, we have standard machine learning models, but the prerequisite to use machine learning models is that you need to have the input ready. That means the features representation must be ready to train your machine learning model. Now, but always it is not mandatory that we will have a trained features with us, like we have a feature set with us. Like if I'm talking about, let's say, the modality of medical imaging like MRI, CT, or PET image, or SPECT image, or endoscopy image. Now we don't have the features from that image, we just have the images in the form of RGB channel, and the image can be of DICOM format, or it can be of PNG format, or the image can be of TIFF format. Like this, we have wide modal different types of image storage. Now, from we don't have any numerical values or such features, we don't have any feature set with us, right? So that is why, especially when I'm talking about medical image classification, it is far better to use deep learning models in comparison to machine learning models. Like machine learning models can be adapted where you have already the clinical features extracted for a particular disease. Like for example, if I'm talking about blood pressure data, blood BP measurement values of a particular patient X, patient Y, patient Z. So I have a numerical values interpreted from the blood test to urine test or whatever test we have a numerical values already for that respective patients, right? Now these are the features derived from a test. Now these features are numerical values, and these features have a set of properties to be in disease X, disease Y, or disease Z. So we have a labeled form with respect to feature values, labeled set with respect to feature values, where we can go for machine learning models. But when I talk about X-ray of a chest, or X-ray of a knee, or MRI of a brain, we don't have any categorization as such directly, because we don't have any features where exactly the tumor is present in MRI, or where exactly the crack is present in X-ray image. So you need to do lots of pre-processing techniques there, and then adapt or identify the respective location where it is. And like this, you need to identify first the numerical feature concept. which is not present with us, but still we need to develop a model which can classify depending on the image. By looking at chest x-ray, it should be able to classify that this patient chest x-ray belongs to pneumonia, let's say one disease. This particular patient chest x-ray belongs to um, um, a COVID, right? Like corona, like something like that, or it is like an pneumonia, or it is like any respiratory issue, right? So that means the image need to be studied, not the features. We don't have any features. The entire image, pixel by pixel, we are supposed to study, and then identify a class, and then identify whether that image belongs to a particular class or not. Now to do this, automatical feature extraction is needed. Like, you need to understand what type of features are present in that particular image, which cannot be done through machine learning models, because machine learning models need features with for it. But for deep learning model has two things inbuilt in it. One is, every deep learning model, if I talk about, every deep learning model will do two things. One is the feature extraction, and then followed by, on these features extracted, it will apply classification, right? So we will extract, we will create, or we will identify the features from that medical x-ray image, and these features are not the proper ones at first stage. So we try to train, or we try to identify the most prominent features which can say that the given x-ray belongs to a disease x, or the given x-ray belongs to disease y, or it belongs to disease z, right? So every deep learning model will adapt two basic modules in it. One is feature extraction, then by followed by an classification module. Now these deep learning models are basically derived from one simple concept of learning, that is convolution neural networks, CNNs, or we can also do using vision transformer models, VIT, which is a concept of encoders and decoders, right? So there are two basic domains where under DL we can either adapt CNN based models, or we can adapt vision based, transformer based models. Now, based on this categories, either it is CNN convolution or transformer. The basically there are two things: either convolution models or transformer based models. So if it is a convolution models, we have n number of DL models again. There are wide sets of CNN standard convolution models. Under them, few of them is like, one of the popularly known model is ResNet, EfficientNet, and so on. Similarly, in VIT also we have wide sets of models. One of them is Win Transformer, and which works on encoder and decoder concept. Like this, we can have different models in deep learning. One is the CNN, or one is the transformer based models. Now let us look at the basic CNN model. Now basic CNN model, it takes an input. Now this CNN basic input is sent to convolution operation. We have here three basic steps. One is convolution, then ReLU activation function is adapted here, then followed by pooling. So there are three basic steps followed in convolution. Now let us look at each one of them for a short. Let's understand how CNN works. Then we'll be looking at what are the standard CNNs and how they differ in properties and how they are adapted in real-time medical imaging, especially medical imaging classification. So now, first, any input, whatever we are taking, the 2D image, the 2D medical image, or 2D image in channels of three, where we call it as a color image, right? Now, these images are sent to convolution filters. So we will be using here convolution layer, we call in general. right? So which generally applies a filter to identify the features, to identify the features. Now it will apply a convolution layer. It gets the features from the original image. Now these features, whatever are being understood or trained or understood from the given image, will be sent to the max pooling layer. And then max pooling, here it is adapted to, max pooling is generally adapted to dimensionality reduction. It is your main purpose here is for dimensionality. It is main purpose is for dimensionality reduction and uh, prominent feature prominent feature extracted, like because it takes okay, let me go one by one. So if I talk about convolution, convolution will work something like this. Like every convolution, that means every CNN in general will have a convolution layer, will have a ReLU activation function followed by an pooling layer, and these will be used in combination. This will be used in combination multiple times. Many convolution layers, many ReLU activation function layers, many max pool layers. Combination is done. Like if I take a block, each block contains a convolution, ReLU activation function, and max pooling. If it is this, I'm taking as a pooling. Such blocks, I'll have many which are connected. Once output is given to the other block as an input. Like this, I'll have n number of layers. Convolution, ReLU, pooling. Convolution, ReLU, pooling. Convolution, ReLU, pooling. Convolution, ReLU, pooling. And then we will be sending that to a dense layer here, a dense layer. This dense layer is nothing but it's already known concept to everyone in computer science field, which is an artificial neural network, which is used here specifically for classifying your data to either binary class or the multi-class. Right now, this artificial neural network, what we are talking about, it cannot understand a matrix. Right, ANN is not meant actually for a matrix. This meant it only for a set of values, a set of values, right? That is why the max pooling output which we are getting as an output of the convolution layers is generally a 2D matrix. Now ANN cannot take directly a 2D matrix, it understands only a vector of inputs. So we flatten this data into a one dimensional array, which we are calling the concept as flatten. Flatten, right? So the 2D feature map, what I'm getting from convolution layers, is converted into a 1D feature map, which is we are calling the concept of flatten. Now this can be sent as an input new artificial neural network. The neurons can be given as directly to the input to the ANN. Now this ANN again uses an activation function like either ReLU or sigmoid or tanh. You can actually play with it. You can have a different sets of activation functions here. Then standard activation function, what we adapt especially for imaging classification, is ReLU activation function. Because we need to have the vanishing, we have to remove the vanishing gradient problem. So we adapt here ReLU activation function both in the feature extraction process as well as in the process of classification. Now after that, this the output of neuron, as we know, artificial neural network, it uses a softmax layer at the end to classify the data into different probabilities in the range of 0 to 1. So it looks at how many multiple classes you have. Depending on that, it will be scaling to different classes, binary class classification problem. right? So like this, a CNN model works. So if you can observe here the complete figure, the combinations of different convolution ReLU pooling, convolution ReLU pooling, this can be repeated in multiple ways. Now this is entirely finally generates a simple 2D matrix which we call as a feature map, right, which we call as a feature map. So that means we are actually finding out the features from an image. Now this image can be a medical image or non-medical image, anything, right? Now since it is actually extracting a feature map, this block from here to here can be said as feature extractor. This can be called as an feature extractor. Once the features are ready, you are actually training your model with an artificial neural network. So this is called, we call as an classification module. So in summary, I can say every CNN model holds a feature extractor followed by a classification model, right? So this combines a CNN model as a whole, if you summarize it. Now, this CNN model, what all it applies, I said, it uses convolution, it applies ReLU, it applies pooling. Now, what is convolution? If I have a matrix like this, a 6 cross 6 matrix, I'll be working on block by block. So I'll consider a smallest block here. Let's say I take a 3 cross 3 block. Now, on this 3 cross 3 block, what is a feature? Is there, do you have a horizontal feature map? Do you have a vertical feature map? Do we have a diagonal feature map? That you can identify through the filters. Now you can have n number of filters here, filter 1, filter 2, filter n, so on, right? Like this we have n filters. Now these filter purpose is to identify what is the prominent feature in that respective block, right? So if I'm talking about image, general image processing, we have only 5 to 6 filters, like 8 directional filters, horizontal, vertical, and with the varied angles, diagonal, right? 8 directions. But if I talk about deep learning, we have to actually extract different sets of features, right? So we have n number of filters in that in our deep learning model. Now, what we are actually doing when I talk about convolution is, I take a block by block processing on image, convolution, applying a convolution operator with the filter 1, and same block I'll apply a convolution operator with filter 2, and so on with the filter 3, filter 4, filter 5. Now, what is convolution is? It is a dot product between the block and the filter. That means corresponding pixels multiplication and the whole sum, right? Sum of the corresponding pixels, right? This becomes your convolution operation. Now, what is the result of this convolution operation? Now, this convolution operation indicates the resultant numerical value, what we are getting. This shows whether what type of prominent feature we are having in that particular block. Now, how do you know that feature is important or not? That is actually depends on what type of filter you have adapted. So for example, here if I take one filter like this, which is on the screen, if I take a filter like this, and to take the feature maps, we can use a stride. So suppose if the stride is 1, then we are actually taking a block by block processing, and block by block processing in the sense that we work on the first 3 cross 3 block and we convolve it. So if you simply look at these two examples here, we are considering a block 100, 010, 001 with a dot product of the given filter. So you can see that most of the values will comprise to 0 because we have most zeros here. And if you dot product it, then the value is multiplied here only with the diagonal values will produce to 1. So its summation leads to 3, right? Similarly, if I go to the next block here, the next corresponding 3 cross 3 block, and if I take a convolution operation and take the dot
Sorry to
product,
interrupt you, madam. May I ask one question here?
uh, yes,
Ma'am, these filter values we create by ourselves, this 1 minus 1, or it is already there? And another question, why these values are only 1 minus 1? Why not
okay,
other?
right. So, are you gone through any medical imaging, basic image processing? Did you study any course?
Not exactly any course, but I have done some work on brain
So,
in
do you
class.
know how an horizontal filter works?
Mm, yes, the filters are applied on a particular block, and by applying the convolution, it getting some edges detected. That
Right, right. So that edges, that values, suppose if I'm taking a filter like 111,000 like this, if I take such filter, so it is actually, we are actually looking at horizontal filter. Like if there is a horizontal edge in that particular block, the value what is given from the dot product will be high. Otherwise, the value will be low, right? So these values are derived. If I talk about image processing, they are derived by default by different operators. As you asked, why we are taking 111 minus 1 minus 1, it depends on it is like an second order derivative filters concept in the basic image processing. So now second order derivative filters will work on the concepts of differentiation between the values. Now these values are derived with the second order derivative functions, which is already derived by many of the scientists like Prewitt operator, Sobel operator, Canny operator, Krish operator. Like this, we have standard filters already in imaging domain, which comprises the values between 0 to higher value. Some values will be minus also in that. Are you getting it? Right.
yes,
So
ma'am.
like,
Yes.
right, so these are values which are already standardly defined in, if I talk about basic image processing. But if I'm talking about specifically deep learning model, these values are not uh, given by it's not by the जैसे कि we have a convolution operator like canny, right? Convolution edge detection operator, canny operator, it has different values in the filter. Similarly, Sobel has different values. Now these are the not the ones which are using. Now what happens is we take in this a randomly initialized values for these random initialized values. Now we try to train the filter with respect to the image, what exactly the feature map is there in that particular block. That means if you look at this image, see, if you look at this particular image, there is a car object, right? Now the car has trees in this background. You have tree, you have a car, you have a road. Now do we have different edges at different location? Now that one is decided through this filter. Now filter in takes actually like in the form of a 3 cross 3 matrix. Now initially randomly here there will be a random initial values. Now it will check whether the first block is having a horizontal edge or not. If it is doesn't there or it is approximate to horizontal filter, then these values are updated. The weight values in the filter are, we call these as weights. These are updated through back propagation in CNN models. Through back propagation um, back propagation in CNN models. Right. So if I talk about standard models, then we have a fixed operators for this minus 1 0 and all. But if I'm talking about CNN, these values are trained by our deep learning model. Are you getting it?
Yes, ma'am. Thank you. One
Okay.
more question. Why these kernels are mostly, we have seen everywhere that is 3 cross 3. Why not 4 cross 4 or
Right.
6 cross 6?
Yes. If I talk about 6 cross 6, 5 cross 5, 7 cross 7, it will be a larger. One thing is that. And the simplest answer for would be like I want to extract the most prominent features from the minimum estimated block. Like if I talk about, actually, this filter has a beautiful generation. When I have a 3 cross 3 filter, actually I'm identifying the feature map with respect to the center pixel. Is my block with respect to center pixel is towards the horizontal direction, or it is towards the diagonal direction, or it is towards the upper direction, or it is towards the right diagonal, left horizontal, like this. The filter concept works on this 8 directions, which is more prominent and more good enough to identify the relationship of that particular center pixel with the neighboring pixels. This can be betterly, efficiently identified in a 3 cross 3 block. If I go for 4 cross 4 or 5 cross 5, every center pixel has to be analyzed with more number of neighbors, right? So in that case it is not that efficient. But if I talk about 3 cross 3, every center is having a relationship with eight neighboring blocks, pixels. So through which the feature map what gets derived in 3 cross 3 is more good enough for further classification. That is why most of the models they prefer to be having a filter with of size 3 cross 3. They use 5 cross 5 also, 7 cross 5, 7 cross 7 is a standard one, the larger. They won't use beyond 7 cross 7. Minimum is 3 cross 3 for better efficient feature extraction. Hope I have answered your question.
Yes, ma'am.
Okay.
Thank you so much.
Okay. So like this, we will be working on block by block. And the first block, we got the feature map as 3. And in the second block, we got the feature map as 1. Right. So now here, the stride can be of 1 or the stride can be of 2 also. Like stride 1 in the sense that first we'll be processing this center pixel, as I told you. Then I'm actually processing the second next immediate pixel. Then I'm next processing the next pixel, the next pixel, and the next pixel, so on. But if I'm talking about stride 2, then I'm actually processing this pixel, then I'll be processing the next most pixel by skipping the next immediate pixel. So the movement of the block is skipped by one position. And similarly, if the stride is 3, then I'm skipping 2 positions and going to the next block, like this. But we generally fix the stride to be 1 because we want to extract the most prominent features from the entire image, which can be done only when you want to process, when we will process every pixel by pixel, right? That is why we will not skip such positions. We will extract the feature map with respect to one. What is the feature map in this direction? What is the feature map in this direction? What is the feature map with respect to all the directions for every pixel? So we try to fix the stride to be as one in most of the standard CNN architectures. We will not go for stride as 2. So like this, we move on the stride and find out the feature maps. So like we process the entire given 6 cross 6 image. So you can see here the feature map is derived here, which is again a 2D matrix, but the size got reduced here to 4 cross 4. We had an image size of 6 cross 6, but the output is 4 cross 4. This is because, as I told you, the feature filter works with respect to center. That means first we are actually processing this pixel with this pixel, what is with respect to this pixel, what is the orientation of its edge, right? Then I followed this, then we followed this, then we followed this, so on up to this position. That means I have looked into the relationships of these pixels alone only. That is why we got a matrix size feature map of size 4 cross 4, not a 6 cross 6, right? But in general, we have to get the feature map of size 6 cross 6, not 4 cross 4. So for that, that means I need to look at this position also with respect to these angles, right? And similarly, this corner pixel also with respect to the three orientations, right? So this I have skipped right now, the ones which I'm getting a feature map over in this place, right? So to generally CNN does like this, and it avoids the corner pixels because the center concept is adapted here. Now I want to have the feature maps with respect to corner pixels also. That is why we do a concept called in convolution called as padding, which means I add an extra row here on all sides to extract the feature maps of the corner pixels as well. Now, padding can be zero padding and the padding can be duplication padding. So we adapt a padding concept to extract the features of all the pixels. So when I do padding with zero, the feature map size will be 6 cross 6. That means for every pixel in the original image, I have extracted the feature map. Like the values we got here, 3, -1, 0, -2, and so on. Like this, I can extract all the feature maps of the given image. So always apply padding whenever you do a convolution based feature extraction. Now, like this, will be doing multiple filters. Here we have shown only one filter. Like this, we have multiple filters. Now, as we have a multiple filters, obviously I will get multiple feature maps like this. So if you have 50 filters, you will get 50 feature maps. If you have 100 filters, you will have 100 feature maps, right? Now, again, this feature map size is again, as I told you, if the padding size, if I add, the feature map size is again going to be a 6 cross 6, right? So in this example, and in real time we will have the generalized images of medical domain. If I talk about specifically, it will be around 20,000 2000 cross 2000 or 8,000 to 1500 into 1800, something like that. Like that, we will have the size of the medical image, right? And that medical image into three channels RGB, so you can understand what is the complexity of that image representation, right? So if I process the filters on this particular entire representation of the image, obviously the feature map is also again of same size, 2000 into 3000 into 3, right? Now this further processing again into the next convolution, again into the next convolution, as it is shown in this particular run. As you can see, convolution relu max polling, convolution relu polling, convolution relu polling, so on. Then finally we flatten it, right? So when I go for combination, repeated the combination, obviously your feature map, if it is of size larger, it will take too much of time, right? So that is why the pooling tries to help to extract prominent features and reduce the dimensionality, to extract the prominent features and reduce the dimensionality of your feature map. So that is why here we apply a two stride. Now what is the back pooling is, like suppose if you have an image like this, then I'll be taking a 2 cross 2 block at every interval and take the maximum of this, right? So suppose if my image is 200 cross 200, I will be moving on every non-overlapping blocks. That means I have processed to first 2 cross 2 block, then I will process the next 2 cross 2 block, then again I'll process the next 2 cross 2 block, like this, right? So my image of 200 cross 200, it will now reduce to 100 cross 100 because of this pooling, max pooling. You can apply here min pooling, you can apply max pooling, you can apply average pooling. In practice, we apply max pooling for better results, right? So like this, we can reduce the dimensionality of the image, and then further we can go for further feature extraction of that image. And finally, the features what we are getting after the feature extraction, that will be like the matrix what get generated as an output of this last layer will be holding the most prominent features which can derive our car, the input as it is shown here, right? So now those are most prominent features which can describe car efficiently. Then those features can be further used for dense layers to classify the model, right? So like this the CNN works. Now this CNN um, can be adapted in wide variety of architectures. One of the standard architecture is VGG. You can see here these two are convolution layers, then we have a max pool layer, then again two convolution, then again a max pool, then again three convolutions, then again a max pool, then again three convolutions, then again a max pool, then again three convolutions and again an max pool. And after this we are actually here doing a flatten. Now after flatten we are sending to fully connected layers. FE stands here for fully connected layers, and this further classifies into number of classes. So this, from here to here it is feature extraction, from here to here it is feature classification, right? So this is how a generalized CNN model works. Now if I'm taking this particular pattern flow, then it is called as an VGG standard architecture model. Like this we have n number of architectures. Now this VGG model will hold around these many number of parameters. Now what are parameters? These are called trainable parameters to train your model. Now every VGG architecture, what I'm showing here, every CNN architecture will depend on the complexity of the model. As we already know in general, like if I have to say this model is efficient, we'll be looking at two things. What is the time complexity? What is the space complexity? So similarly for this architectures also, we will be looking at how many trainable parameters are there, because on which only your time depends and your efficiency depends. So trainable parameters should be less. It cannot be large. If how many, depending on number of trainable parameters, the model becomes too complex. Right now, these parameters can be identified with a one standard formula, which is a filter height, filter width, number of channels, number of filters, plus the biases, because every filter will have a one bias. Right. So using this formula, we can find out how many parameters every layer can take. Now, if I calculate all such parameters for VGG architecture for every layer by layer, then finally I'll be getting around 138,357,544 number of parameters. Now, if I compare all the standard CNN models, we have some standard model called LeNet, which takes around 60K parameters, AlexNet, which takes around 61 parameters, VGG 16 takes 138 million parameters, VGG 19 takes 144 million parameters. Similarly, ResNet 152, that means how many layers are we taking, is 152 number of layers, which takes around 60.2 million parameters. Right, like that inception takes 55 million parameters. We have one standard architecture of CNN, which is called dense net, exception net, mobile net, and so on. So all these are based on base concept of CNN, feature extractor and classifier, and base concept adapted here is the convolution layer, convolution based feature extractor. Now, if you can observe here, the mobile net is taking very less number of parameters, like only 4 million, 3 million, 5 million. What is the need of this? Why should I don't use the higher dense models? Because if you look at DenseNet 264, it is very deep neural network. It can extract more prominent features compared to mobile net. But in real time, do I have to go for a standard with newer number of layers or a standard with less number of neurons? Depends on the application. Exactly. Like if I'm trying to deploy this classification model on small sensor device like a camera, then if this camera is already having low memory device, It's like a memory constrained device, right? So if I deploy a DenseNet 264, it cannot be handled with some small IoT device, because it needs high processing time and high processing memory, so which cannot be handled with smaller devices. So every smaller device should hold architecture for classification, but with less number of parameters. So mobile net would be a suitable one to give you the efficient classification with minimal number of parameters, and it is adaptable to any type of medical device, any device, actually not medical, any device. Now in medical domain we need faster results. Within fraction of seconds we want to have a prediction, because if I take even a fraction of second delay in identifying the patient behavior, then there could be some big mishappening to the patient, right? So I have to handle in few seconds. But that monitoring should be done continuously, right? So we have to use or deploy the models which are in lightweight with respect to parameters, and it should be also efficient to extract the most prominent feature maps from that particular patient. That is why we look at the lightweight models with better efficiency in medical domain, and also adaptable to the real time. Not only the simple small lightweight, but it is not suitable for a particular disease, then there is no use. So you have to look at multiple constraints. So regarding this, we have certain applications already developed using deep learning for medical image classification. For few publications, few of my students have already done. Let me show up them. So one of their such application is an ensemble model to detect COVID-19 and pneumonia using chest X-ray images. So here we have actually using chest X-ray of a patient. So like this, in this architecture, you can see we have the chest X-ray of a patient indicating the pneumonia, indicating the images of normal patient, and from the COVID patient, we have a three collections of chest X-ray images. Now we have ensembled model used here. One is the efficient network and the mobile network. So these images are passed through efficient net CNN model, mobile net CNN model. Whatever the feature flatten data, 1D feature map, whatever it is generating, 1D feature map, whatever it is generating, we have concatenated here and then used a set of fully connected layers for like this set of fully connected layers for classification to COVID, normal, and pneumonia. So you can see here in the last table also the set of accuracies for class wise accuracy. So we can see that COVID-19 is having around 98% accuracy, pneumonia 95% accuracy, and normal we got around 89% accuracy. So this is the model which is for chest X-ray classification using ensemble model. Similarly, we have another paper which is used for um, bleeding and non-bleeding endoscopy image classification, wherein we adapted a mobile net architecture, the standard CNN architecture, which is named as mobile net. And since here, if I look at specifically about the medical domain called wireless capsule endoscopy image, here the images are something like this. Like we take the endoscopy images, where the images might look like this in the form of A or B. A is a category of endoscopy images with the blood floating, and the B is now non-blood floating. So if there is a heavy blood floating, then immediate action need to be taken on the patient. And if there is no blood floating, then we can actually relax here. So immediate action need to be taken here. So that here for this particular classification, we have adapted an model called as mobile net followed with the color, because we have a dominance of red color. If you look at the sample images in A, the red dominance is more, but in the sample of B there is no much dominance of red. So red component is playing a wider role in classifying these two category of images. So we adapted a model to extract the color histogram a color histogram, and also adapted the features of the both the images, whether it is bleeding or non-bleeding, through mobile net by processing block by block as a convolution layer model. Now then, after that, we have flattened them and concatenated both the features. Then we applied a set of dense layers, which is simply an artificial neural network, and we applied a softmax function over here to classify bleeding and non-bleeding. Right. So this is one such application. Another domain is different. So I have shown you two different domains where the CNN have are being adapted. One is X-ray image classification. Another one is endoscopy image classification. Similarly, we have adapted here another combination of uh, ensemble model for classifying the COVID-19 image with the non-COVID-19 images, again using the same chest ray X-ray images. Here the modality, here the mobile net is adapted again with an enriched features classification here. So here we adapted the basic pre-processing. One is the standardization, augmentation, and all. Then we have extracted the features using CGAN. Then we applied a modified mobile net approach, and then we applied recurrent neural network for classifying the into three categories by finding out the um, count score, actually moderate, severe, or critical. Like this, we adapted one paper in the field of same medical image domain with adapting various sets of CNN models. So these are the different accuracies of that respective paper. So you can see there is a higher accuracy for the proposed model. Similarly, there is another paper here which is also adapted with the same X-ray image classification, COVID and non-COVID. So again, here we adapted an custom CNN model, a deep neural network we have developed by our own, which have named it as an SRC2. So we adapted or we created our own CNN architecture. Our main target in this paper was to reduce the number of features and give the same accuracy as the standard architectures, which are having high parameters and giving the same accuracy. We adapted a small CNN model with less number of parameters and proved that the accuracy is going to be the same. Similar accuracy we can obtain with a lesser weighted, lightweight model of custom CNN. So with this, we have found the accuracy also. So you can see the proposed model had only these number of parameters, but the existing models had very high number of parameters, and we tried and we achieved an accuracy of 98% all with our own feature extractor model and our own classification model respectively. So like this, we adapted most of the deep learning models in real time to adapt the classification in with better accuracy. Now, but even then, though the accuracy is high with the deep learning models, there are certain challenges left out in deep learning models as well. That is data set requirement. If I look at the second point here, deep learning models requires large labeled data sets for training. That means you need to have huge data set for training the model, right? And accuracy is always high accuracy is obtained because you have a large data sets, right? But in real time, do we get really such large data sets specifically in medical domain? Is not guaranteed, because in medical domain, particular diseases, let's say cancer, right? So cancer also have different types of cancer. Now some types of cancers are very rare, where you don't have huge sets of samples, you have only samples in the form of sets of hundreds, and other cancers data sets are in millions. Now how can you train a model wherein you have only less number of samples with the comparatively analysis of other cancers which are of very high, right? So a model will be biased if I train the data set directly like that, even with the deep learning. And deep learning is a requirement that the data set should be high, right? And the due to which the problems gets into encountered, like data imbalance and adversarial attacks can happen like that. We have lots of challenges here. Now, to overcome these challenges, if I summarize the challenges, that is, one is the requirement of larger data sets, data imbalance, and everything is black box. Nobody knows what is happening in the deep learning, though we know the architecture layer by layer structure, but how exactly, I told you how the neuron is updating, how backpropagation is used, all these things, how the values are updated, how the model is trained, this nobody knows exactly. You know the concept, but what exactly the value is there in after weight updation, this we cannot know, right? So this is very high computationally costly. So that is why we try to go for the next future in classification models, which is quantum based machine learning models, like QML, you can say quantum machine learning, right? Now, why quantum machine learning? To speed up, it is, you can do the parallel processing in quantum processing. So that means the advantages is, one is the high dimensional data can be understood here, you can do huge parallelism, and you can have the quantum inspired neural, you have to use the quantum inspired neural networks obviously, but it will give you the better efficiency if they are developed in future. Right now there are very limited quantum machines available, due to which we are facing the hardware challenges as well as the software challenges. But in near future, like in within 5 to 6 years, I hope that we might get a better quantum model which can be deployed in our servers, and we can use it for our local machine. I hope this will happen in next to 5 to 6 years, right? So let us look into what exactly in the quantum we have done. One of my PhD scholars thesis was going on this, and he adapted, we try to adapt the class quantum machine learning model for feature extraction or for the classification. So we looked at both the cases. For feature extraction we have used quantum machine learning model, and for classification also we have used the quantum machine learning model. Now, under this, we have some relevant architectures. Because the time is less, I would like to go through the main concept. So, we have published one paper here, which is a hybrid framework for respiratory lung disease detection based on classification CNN and quantum classifiers from chest X-ray. Now, same data set we have used for used in classical models also, like we have adapted the custom CNN we have used on the same data set, and we have used the ResNet model on the same data set. We have used MobileNet on the same data set. Now we have used the quantum machine models also on the same data set. Now, under quantum machine learning models, you can see in this architecture module 1 and module 2. Under module 1, which is a feature extraction, wherein we applied a custom CNN, like existing CNN feature extractor we have used, model based custom CNN we have designed here, and then we followed with an classification model under quantum. So we have converted this feature extracted, which is flattened data from the feature extractor. We transform them into qubits, which is required for quantum processor. Now these qubits, we have an encoder there. We have used the quantum classifier MMS and MSMS. We have designed two different classifiers here due to, and then we followed with the softmax. We classified into the pneumonia and the non-pneumonia, cancer and sorry, COVID and non-COVID. So, like this, we have adapted the quantum classification. The model can be said as an, for feature extracted, we used custom CNN. For classification, we adapted quantum classifier. So it is like classical and quantum hybrid model. So with this, we had a better efficient accuracy, which is about, if you can see in this figure, we can see that CCML MMS and CCMSMS, we can see the accuracy for here, it is around about 98%, right? Then again, we have another paper here where we have reversed the concept, hybrid QCC. In this, we have done a hybrid framework for automated respiratory disease based on quantum feature extractor and custom classifier. We have reversed the process. So first we applied here quantum machine learning for feature extraction, and for classification we adapted the custom classifier like dense layers, artificial neural network classifier. So here the feature extraction is done through quantums, and once a quantum has given our feature map, those are converted back to the from qubits, it has been converted back to a numerical value, then we applied to the custom classifier. Now with this again we have a good enough accuracy again. So you can see here with the q depth 4 matlab the qubit 4, if we have a qubit representation, that I will take another lecture if it is possible in future develop FDPs or so on. So I can explain there what is exactly the quantum qubit and so on. So if we have used here quantum 4, we got the accuracy to be as 86, 97, and 88 respectively. And if we have used quantum 6, we got the accuracy about 87, 97 and 88.9 respectively with respect to F scores and precision, sensitivity, accuracy, and so on. Similarly, if the same data set is been adapted with normal mobile net, VGG, ResNet, and so on, this is the trainable parameters. You can see the parameters are many for classification models, but if I am talking about the parameters in quantum machine model, the parameters are less, and the training accuracy is almost the same. I won't say we have got FE best, most the best accuracy, but we got the similar accuracy like in classification models, but the processing time, the parameters were less here in compared to deep convolution neural network. This is another comparison with the literature. We had a 41 reference and so on with the various standard deep learning models again. So we have seen the different ways here, the different feature extractors, like we adapted ResNet for feature extraction and proposing our own classification model, and custom classification for feature extraction, and we adapted one quantum machine learning model. With that also we tried to we got a better accuracy in comparison to the standalone approaches. So you can see here with the custom, our own custom model for feature extraction and our own classification model of quantum, we reached accuracy about 96%. And in the standard deep learning models also we have a similar accuracy. I won't say that we have bypassed the accuracy, but we got a similar accuracy even with the quantum based models. So in future we can adapt quantum lane based models with if case the hardware is available to everyone, and if the price gets reduced, the usability prices gets reduced, we can adapt them and we can do the even more better efficient classification with less number of samples as well, which is the main drawback in the deep neural networks. We need to have large sets of data sets, huge sets of data sets for a particular medical image domain classification model. We can restrict it and we can adapt quantum machine learning models as well. So this is a overview of how what is a CNN, what is a medical image domain, and what are the types of medical images, and what are the models we can have in standard deep convolution neural networks, and what are the limitations of it, how they can be used for better efficient calculation of efficient classification of medical image domain. And then we can use the next future we can try to use for cloud based quantum machine learning models, and then we can go for regulatory considerations in future health AI and so on. So this is what I have discussed today. What is the basic of CNN, then followed by standard architectures, the drawbacks, and then how they can be adapted in real time, and then how quantum are able to give better accuracies as compatible to the standard deep neural networks. Thank you, sir.
Hello, ma'am, please share your
Yes.
email ID.
Yeah, sure. I think on the first slide, I have my email ID. Yes. It's rajitha@mnnit.ac.in.
Any queries?
Any questions from the participants?
I think there is no queries anymore.
Thank you, ma'am. Session was very very interesting and nice. And you started from basic concept and then you went towards your paper. So basically, quantum computing and all these things are now very new nowadays. So if we could do this kind, I'm working, ma'am, uh, in my lung diseases, and
Okay.
I am using the CNN. But this novel idea we can use, as you suggested, quantum computing, and I will contact you about your work.
Yeah, sure. I'm available on internet. Like through email, you can contact me, or through on my phone also you can contact me. We can have a discussion on it later.
Okay, ma'am. Thank you so much once again.
Okay. Okay. Okay, sir. Then I
Thank
hope
you,
I
ma'am.
can leave.
On behalf of the organizers of this faculty development program, I extend my heartfelt gratitude to you, esteemed speaker, Dr. Rajitha B, for delivering such an insightful session. Ma'am, your session on the future of medical image classification, deep learning, and quantum was nothing short of enlightening. You not only share your deep expertise but also ignite curiosity and excitement in all of us about the future possibilities in the domain. The way you simplified complex ideas and connected them with real-world application has left us motivated and eager to explore further. Once again, thank you so much, ma'am, for enriching us with your knowledge and wisdom.
Thank you, sir. Thank you.